All right, hello everyone. I'm Derrek Hibar. So as Jason mentioned, I'm going to be talking about utilizing big data sets in imaging genetics. I pose the problem or I pose a series of general research questions and as a way of introducing the data sets that are available that are at your disposal for your research questions. This, oh.
Yeah.
So I think you have a bit of background already about why you would want to do this, but just as a reminder, why we would want to study brain imaging and the genetics of brain imaging. I think it separates into three sort of very general domains. Your research questions do, I should say. The first being that we can use genetics to understand the brain biology. And then the other two are related to understanding disease and other phenotypes, behavioral phenotypes in general. And the value of those, I split them separately into characterizing known disease variants. So if you, in your own research you find something interesting and you want to further understand how it affects the brain and brain structure. And then also the third domain is identifying new disease variants, which is using a brain measure as an endophenotype in order to discover new variants that are related to a disease. And it falls along that spectrum. And then I think, am I able to point? I don't see. Okay. Yeah, so I mean, here along, this is a representation of disease, but obviously it could be anything. It could be schizophrenia, it could be Alzheimer's disease. The imaging phenotype that you ultimately choose could also be anything. I think this is an imaging measure. This is an imaging meeting, so that's clear to everyone. It could be structural imaging, diffusion imaging, fMRI. But when it comes to DNA, there's also a choice. I think so far we're pretty familiar with the concept of a SNP, a single nucleotide polymorphism, that's been mentioned by JB and Roberto both. But there are obviously several different types of variants in our genome beyond just SNPs. Rather than introducing every single type of structural variation in the DNA, I think the concept that's most important is how frequent they are. So single nucleotide polymorphisms are very frequent, and then there are other types of structural variations in our genome that are less frequent, and that has a big influence on the types of analyses we can do and various trade-offs. So what does that look like? On the x-axis here, you can see the frequency of a given allele from 10% of the population having a variant and all the way to a very rare, being 1 in 1,000 that has a given variant. Those that are most frequent are the SNPs that we've mentioned, and that's the arena of GWAS. That's where most GWAS variants live. And then in the very rare space, those are sort of private mutations almost, or those that occur very rarely in the population in general. And the title of the slide is Life is Hard, because the other key component of this is there's a trade-off between how rare something is, how common it is, and then how big an effect size you can expect. As JB mentioned, once you have proper sample sizes, you start to realize in a very large GWAS that the individual effect size for a given SNP is quite small. So in order to find a believable and replicable variant in a GWAS, you need a very big sample size. And conversely, in a rare variant analysis, because the variants themselves are rare, you need to have very big sample sizes in order to find the variant in the first place. There are some unique aspects to this as well. In the rare space, you can also do family-based analyses, and I think you'll hear from David later about family analyses and then possibly also very large population studies in rare variant analyses. But the trade-off generally falls along this diagonal line. And I'm going to mention here these two can be tackled with whole genome sequencing, and then this one really falls in the domain of genome-wide genotyping. So those are SNP chips, which I think maybe you've heard of before. Just they capture common variation across the genome, and they're very cheap to apply. So I guess the first research question in general is, what if you have, say, an Alzheimer's disease mouse model, and one of the variants is a missense variant, a missense SNP that influences hippocampal volume. But it turns out that that exact gene variant is very rare in humans. So what can you do if you want to sort of extend your findings from your mouse model to humans? That's the first general question. And again, if you notice, it links on how frequent the variant in question is. The answer in green here, which I hope you can see, it's not, okay, yeah, is that you first need a large data set. And you can see in each of the answers I give for the questions, they all hinge on having a large data set to answer your question. But for the rare variants, it's good to have access to whole genome sequencing data. And in fact, there are a few data sets, the biggest one being the Alzheimer's Disease Neuroimaging Initiative, which, if you're not familiar with it, it's a really great resource to get started, especially if you're working in the Alzheimer's disease or aging space. It's quite a large data set, 1,900 patients with longitudinal imaging, and it also covers multiple modalities, so it has structural MRI, DTI, and PET, in addition to blood-based biomarker, serum and plasma measurements, and including CSF. It has a combination of genome-wide genotyping in 1,400 people, but the key for answering that research question is that these data have been sequenced, so they have whole genome sequencing in a subset of 800 of the patients, that's a pretty good start, basically. I mean, I think the total sample size for whole genome sequencing is probably not sufficient to do discovery-based analyses, but if you're testing a hypothesis of a single variant from your own research and applying it in trying to replicate it, I think that would probably be sufficient. Again, though, think that there's this trade-off between how frequent a variant is and how often it occurs in a data set. And those that are very rare, you might only expect to find one in every 1,000 subjects, but here we only have 18 subjects. So the number of patients that have the variant of interest might not be very high. In terms of in previous years of this talk, I actually gave a tutorial for processing, accessing, and working with the ADNI genome-wide genotyping data. And those are available online. It's a good introduction for if you haven't run a GWAS before. This takes you through all of the steps, starting from the raw files, performing QC and things like that. And then on the GitHub here, there are some additional resources that might assist you in working with the ADNI data. So another possible research question is, say you work in the cognition space or IQ, and there's some common variant there that you have a hypothesis about, and you want to understand or find if that particular variant is associated with brain structure. Again, in this case, it's a common variant, so it might be a problem that you can tackle with genome-wide genotyping in a GWAS. So you'll need, again, a large data set and access to data with genome-wide genotyping and imaging data, or as you'll see, also access to meta-analysis data from the Enigma Consortium. One relatively new resource for researchers out there is the UK Biobank, and this, I think maybe many people are aware, but if you aren't, this is an incredible resource. It's a population study of 500,000 subjects. As of right now, there is an ongoing process where they're releasing genotyping data on all 500,000 subjects. So people around the world are all currently downloading it. And once the downloads are all complete, they'll release a key to unlock all of the data all at the same time, which is kind of cool. And so that's available data on 500,000 subjects. The number of subjects with brain imaging data is a small subset of that 500,000. It's about 10,000 subjects, but that's growing over time. They do have actually really extensive testing available from clinical interviews and other. My favorite question is the thickness of butter/margarine spread on baguettes. So it's actually extremely detailed, the data that's available. And so if there's a data showcase that you can go and just search to see if they have similar data for your research question. Unfortunately, it's not free to access. There's a fee recovery that they try to get just to help cover the costs of delivering the data. But really, this is sort of unprecedented. So hopefully, you can team up with someone to get access to the data. There are several other projects now that have genome-wide genotyping and brain imaging. This is not an exhaustive list by any means, but there are these are a sampling of them that you might use depending on your research interests. So there are two pediatric cohorts, the PING study, and then also the Philadelphia neurodevelopmental cohort. And then for Parkinson's disease, there's the PPMI data set. Those that are looking at connectivity, the Human Connectome project actually, as far as I know, doesn't have genetic data available now, but there are twin data sets in that you can look at heritability and things like that. And then I just learned about this cohort, but there's not data available just yet, but it's the German national cohort, and it's very similar to the UK Biobank in scope. So I just mentioned a few data sets that have genome-wide genotyping data available. And from Roberto, you learned a bit about how SNPs capture, they tag common variation across the genome, but your particular SNP of interest might not be available on the SNP chip. And the way that you get that data is actually through a process called imputation. That basically takes a set of highly sequenced individuals and then uses the LD structure, the correlation between markers in your data set and in the reference sample to impute or determine the genotype of your own samples for unobserved markers. Previously, there were this was actually kind of a difficult process, and you'd need like a bioinformatics person to help you do this. But there's a new tool that I think is worth mentioning, which is the Michigan Imputation Server. The steps to do this are pretty simple, but essentially you perform the QC locally on your own data, and then just upload it, and it'll handle the rest of the process, which includes phasing the data, imputing it, and then putting it in a format that you can use. One actually nice thing about this imputation server is that there's a brand new reference sample called the HRC, and this is a huge data set, a reference panel of I think 30,000 subjects. And it's not available anywhere else. So the only way you can use this as a reference sample is to use an imputation server. There's the Michigan one, and then there's also one at the Sanger Institute that does a very similar job. But yeah, the only way to get access to this really big sample as a reference sample is through one of these imputation servers. So it's highly recommended. So going back to this, what if you don't want to go it alone? So you had access to UK Biobank and some of the others that I mentioned. Does it make sense to collaborate? And I think the answer is yes. And probably the reasons for collaborating are clear. I think it's already been mentioned once why most research findings are false. One advantage of performing science in a collaboration is that you have several independent sources of data, which can serve for testing and replicating problems, and just in general coming up with new ideas and looking at problems from a different angle is obviously really beneficial for science. To that end, we've formed the Enigma Consortium. I think Roberto has mentioned the original project was started by Jason, the first GWAS of hippocampal volume, intracranial volume. Since then, it's really just exploded into a whole different set of areas in imaging genetics. And really, now it's a platform for collaboration. It's open for proposing new studies, which I'll mention in a second, and how that works. So you've already seen this, but just as a reminder, these were the data from the Enigma 2 study, which was published last year in Nature. It's a GWAS meta-analysis of 8 traits. These data are all available for download, the summary statistics on the Enigma website. So several methods that are being developed allow you to use these statistics for your own research and test hypotheses. And also if you just want to look up SNP results for a SNP of interest, you can use those data as well. This was a follow-up study recently, basically looking at hippocampus and ICV in collaboration with the CHARGE Consortium, which is a similar consortium to Enigma, though it's slightly more closed in terms of access to the data. A new project that we're working on now, which is ongoing, is to perform a GWAS of cortical thickness and surface area. And it's maybe not a surprise that cortical surface area and cortical thickness are highly heritable. You can see here the regions in yellow are most heritable, and those in red are still at a level of 0.6 or and above. So the brain in general is heritable, and these particular phenotypes are very heritable as well. And so that's basically what we chose to target in this new project. Currently, we have preliminary findings shown here. We have about 18,000 subjects, 15,000 of which are healthy controls. We performed free surfer parcellations and extracted surface area and thickness in all 18,000 subjects. And then we imputed the GWAS data to the 1,000 Genomes Reference Panel, and then performed an association across brain regions. This is all preliminary, so I don't want to talk about specific SNPs that we've found thus far, but there does seem to be a general pattern where a single SNP affects multiple regions. One thing that we've found in general is that SNPs, we've found several SNPs that have a strong effect in surface area, but not in thickness. And here's another example of that. So just to emphasize, this project is all preliminary and it's ongoing. The first round of meta-analysis is complete, but as a second replication cohort, we're still looking for collaborators. So if you have access to data and want to participate in this study, just email the Enigma address there. But I mentioned, so that's an ongoing GWAS project, but I mentioned that Enigma is more of a platform where you can propose a study and execute it with collaborators around the world. This, I'm not sure if you can see it very well, I'm sorry, but this is just showing all of the genetics projects in Enigma, and there are quite a few. I wanted to just highlight two examples. This is a new project from called the Enigma CNV project. Basically, Ida Sunderby in Norway proposed this project idea to look at variation in 15q11. It's a relatively common CNV, and she proposed it to the Enigma consortium, and it was approved. And she quickly obtained data from 14,000 subjects to perform her analysis. And basically, she proposed to test the association between this CNV and intracranial volume. And another example, this is work from Frank Sun from Carrie Bearden's lab. They have a pretty sizable cohort of patients with 22q deletion syndrome, but their individual sample wasn't really enough to see significant effects, and they also wanted to increase their sample size and improve the robustness of their findings. And just here is, you can see actually they've amassed quite a large sample, so 466 cases of 22q and matched controls from 10 sites. So they essentially had a data set, and they proposed a new project within Enigma, and were able to find 9 more research sites around the world. And they've done a combined analysis looking at the relationship between 22, just basically the effect of diagnosis in cortical thickness and surface area. The effect sizes are shown here. And they've done other stuff too. It's a really great project. And I mentioned before that the meta-analysis data from Enigma GWAS projects are available online. If you go to downloads, there's a specific area to download each of the project data from Enigma. Some of them require that you fill out a small form just saying what you plan to do with the data, and that you won't do anything crazy with the data. Crazy meaning, ethical things. And then also, though, there are tools in development in Enigma, on the Enigma site, including doing things like enrichment tests. So I think that Roberto covered some of those that you might do. So testing the genetic correlation between a phenotype of interest and brain GWAS. You can do that here on the Enigma site just with the drag and drop interface. If you're not so great with coding, this is a great resource for that. And here's basically just an example that we did in our recent ICV GWAS paper using that tool, which is just to look for other traits that have GWAS data available, upload those to the Enigma website, and then test the correlation with ICV. And in several cases, the things that we found were obvious. So child head circumference is associated with, is correlated with ICV, and some other cognitive traits as well. This is just a slide describing some other features of the Enigma Viz tool, and Jason will talk about the meaning of some of these functional annotations and how they might add to your analyses. But this is something that you can also get from the Enigma Viz tool. And I wanted to end by saying that in the Enigma consortium, all of the projects that we do, we generate code that gets distributed to sites, participating sites around the world, and all of that code is available on the Enigma GitHub. It's a really great resource. If you don't use Git or GitHub, it would be really worth your while to learn how to use it. But if you don't want to learn how to use it, there's also just a website interface where you can download scripts and things like that. And so just to end, thanks to all of the Enigma collaborators and the Charge Consortium, and then also acknowledging the NIH Big Data to Knowledge grant that we received. Thanks.
Thank you. Any questions for Derrek? Would you mind going to the microphone, please? And we'll have someone come up.
Very nice talk. And I just have one question. So most of us Enigma studies focus on the structural MRI. I'm just wondering, is there a push to collect more functional MRI, like resting state as well? And if that's the case, what's the challenge of doing big data imaging genomic using functional MRI?
Yeah, so yeah, that's a good point. I think I covered mostly just the structural MRI stuff. There's another project that I didn't mention, which is looking at GWAS of functional anisotropy from diffusion tensor imaging. When it comes to functional MRI, we were less convinced that the the imaging-based measurements from functional MRI could be combined in a similar way across sites. There seems to be pretty significant site effects, study effects, just by scanner. But there are efforts within Enigma to develop a harmonized analysis protocols for resting state specifically. There's not a current effort for task-based MRI, fMRI tasks, but there is an effort underway for resting state. But really the challenge is first to convince ourselves that we can combine the data across sites. Yeah.
Next question.
I don't think it is. No. Yeah.
So you mentioned cortical surface area as well as cortical thickness, and I was wondering, and the heritability of those.
Yeah.
I was wondering, how does gray matter density fit in to that?
Yeah, so I think, gray matter density is really similar to volume. So what we did essentially was, there are three types of measures that you can get from FreeSurfer for just directly that it calculates, and surface area, thickness, and volume are one of them, are each from FreeSurfer. And our thinking was that volume is just a combination of thickness and surface area. So we focused on those, and there's previous work showing that they are genetically separate. They have separate genetic determinants, and the work that we've done so far does seem to indicate that there, while a particular variant might have an effect in several regions within surface area, it doesn't seem to have an effect in thickness and vice versa. There isn't necessarily, like, in terms of gray matter density as calculated by like SPM or VBM or something, there's not currently an effort to look at that, though I think the results would be similar to a volume-based GWAS, yeah. Yeah, more localized. Yeah, that would be if it was voxel-wise within a region. But I was just thinking ROI. Yeah.
Okay, great. Let's thank Derrek again.